Okay, so if we don't, so we would be happy enough to make our protein here. So this is the end of our protein sequence. Okay, but if we don't, and we're using BAMH1 to clone, if we don't actually add this extra A, then our GFP that we want to make stuck on to our protein won't be read in the same reading frame, and therefore the reading frame will be shifted and it won't make the GFP protein. So if this A of the ATG, instead of, if we didn't add an A in here, would be shifted over to here, the next reading frame triplet codon would be TGG, and that doesn't code for a methionine, and then the next triplet codon will not code for a valine, and you will be totally out of frame and you're not going to make that fusion protein anymore. So we add an extra A to essentially ensure this correct reading frame to make our protein sequence, followed by the green fluorescent protein sequence in the correct frame. So when you make the protein, you actually make both proteins in the same reading frame. And so this is what we have to do all the time, is after you've decided which restriction enzyme sites you're going to use in a multiple cloning site, you have to be aware of these reading frame issues that depending on the restriction enzyme sites you choose. And you have to add bases if the restriction enzyme sites are not in frame. So you can see, for example, this EcoR1, which is a very common restriction site to use in cloning, is GAA-TCC. This also doesn't follow the same reading frame. So you would have to also add an extra DNA base if you want to continue the same reading frame. So these are all things we have to do normally when we clone genes into vectors. Okay, so how do we clone this? Say we actually wanted to clone this TP63 gene in with restriction sites. Okay, so we know in the lab we're not doing it that way. In the practicals we're using this TA cloning, but say we actually wanted to use the normal way of cloning with restriction enzyme sites. What would we do? How would we do it? Okay, so say we're using this is the multiple cloning site of the plasmid, so it's the same GFP plasmid that we were having a look at. So anyway, a multiple cloning site of a plasmid. We're going to use Hindi3 and BAMH1 as our two ends that we want to engineer into the bit of the TP63 gene that we want to amplify. So how do we do it? So if it doesn't actually contain these sequences for Hindi3 and BAMH1, we actually use PCR to introduce these new sequences on the ends of the gene that they weren't before. So you design an oligonucleotide primer, and the next slides will explain that. So you engineer a forward primer that isn't just the start of your gene, but before the start of your gene, you actually have the 6-base nucleotide sequence of the Hindi3 restriction site, and your reverse primer isn't just the end of the gene that you want to doesn't just border the end of the gene you want to amplify. It actually has the 6-base pair DNA sequence of the BAMH1 site tacked on the end. And so once you use these primers in a PCR reaction, all of your gene that you amplify with these forward and reverse primers will also have the restriction sites tacking on the end, because they're going to be made using these same primers that have these restriction sites that you've synthesized already. Okay, so let's have a look. So we've got, here's our TP63 gene. So we need to design these forward and reverse primers. So obviously here's the forward primer. So this is the start of our TP63 gene. So this is our ATG start codon, codes for a methionine. So then every other triplet codon after that will code for the protein sequence. So this is then the protein sequence of your TP63 protein that you make from that, from that gene sequence. Okay, so you need to have, like you've looked for in your computer pracs, your forward primer, although it wasn't at the start of the TP63 gene, this one is, it essentially has a small sequence that is homologous to the TP63 gene. Okay, so that's what the stuff is in red. So you can see that it's just homologous sequence to the TP63 gene. So we need to have that, because then the primer doesn't bind to the boundaries of the gene that you want to amplify up. So you always need that. Then what is new is that you actually, on these primers get made by a company that synthesizes DNA. Okay, so you tell the company what you actually want synthesized, what sequence your piece of DNA is going to be. And so you can tell them you want a lot of stuff. So in this case, for the forward primer, you tell them you want a Hindi 3 site before the start of your complementary bit of your gene sequence. So here's the Hindi 3 site. And then you also need an extra bit of DNA bases on the end after your Hindi 3 site, because the restriction endonuclease is a big protein and it needs some space to sit on the DNA. And so you always include it, and they're not, it doesn't really matter what these bases are in green, but just that there are some bases after the restriction site to make sure that when the restriction enzyme finds this site in the DNA, that it actually has some space to sit on the DNA. Okay, so that's essentially the sequence that you would then order as your forward primer. So you can see the bit of DNA that's complementary to the gene you want to make up. Then you actually your Hindi 3 site, and then a few bases that ensure that the restriction enzyme can cut the DNA. And that would be the same for your reverse. So your reverse primer will have the reverse complement of the end of the gene that you want to amplify up. So remember, the primers are always the boundaries. Okay, so whatever you want to amplify up in between the primers is the identity of the complementary bit of your primers. So the reverse primer, if you remember from your computer prac, is always the reverse complement of the coding strand of DNA. So it would be the reverse complement of the coding strand. Then you have your restriction site that you're going to use at the end. So your, in the case BAM, so GGA-TCC. And then you have again another few bases at the end of that restriction site, just to make sure that the restriction enzyme can cut. And so each time you use these primers in PCR, then you're going to make your gene plus all of this sequence on the ends of the primer, and it will always be there. So each time you do the PCR cycling, then you'll continually make up the gene with these flanking new bits of DNA that weren't part of the gene before. And so that's essentially what happens, and that's what we do. What we do all the time is that you then make this population of your gene that you're trying to insert with the engineered. Now you know what engineered means. So the engineered sites at the start of the gene and at the end, the two different restriction sites that you then continually make up when you do a polymerase chain reaction. Okay, so and and then obviously you again. So I don't know why I've included this one again. We obviously also have to make sure that if we're making a fusion with the green fluorescent protein, that we have this extra DNA base in here to make sure that we account for the reading frame being conserved. So these triplet codon being conserved, and we can make our protein and the green fluorescent protein tacked on the end of it. Okay. So the last few slides, I'm just going to talk a bit about how PCR works and a few examples of how you can use PCR in different fields, and just a final small bit of discussion essentially about your PCR assignment. Okay, so you will have already started to use PCR. So you've done essentially 2 PCR reactions in your module 1. You did your first PCR reaction where you PCR'd up TP63, or the bit of the TP63 gene, and then you're doing another 2 PCRs to introduce mutations into the TP63 gene. So at this point, you have to start to understand how PCR works if you don't already, because this is also the basis of the assignment that you have to explain. Okay, so the point of PCR is you use a step at the beginning of the PCR where you break open the DNA strands. Remember, DNA duplex can't do anything with, and it's the same with PCR. You need to separate the 2 strands of DNA, so then your primers can anneal. Okay, so this is your forward and your reverse primers that will anneal to the bit of DNA that you want to, the boundaries of the bit of DNA that you want to make up. And so obviously over you need a polymerase that's going to extend, make a new piece of DNA across here, and using that primer, and then a new piece of DNA across the top, which uses the reverse primer. Then you end up with a lot of DNA that is bounded by the two primers that you're going to make. And so you essentially go through these cycles of opening up the DNA, annealing the primers, making a new DNA strand, extending the DNA strand, and then going through the same cycle over and over again until you get lots of population of your gene that you want to amplify up. Okay, and so that's essentially the steps that you also have to explain in your PCR assignment, so the basic steps that we've just said with the PCR reaction. So you have to kind of explain it in a more detailed terminology using figures. Figures are always useful to show that your understanding of things. If you make figures yourself, you never have to reference them. If you're using figures from, say, a textbook or from a research paper, you must always reference figures, or else we assume that you made them yourself. And if you didn't make them yourself, then that would be where plagiarism would come in. Okay, so then obviously step one, again, of the PCR reaction is what I've just said. So you have to open up the DNA. Step two is annealing. In the case of PCR, it's annealing the primers. Extension is where the DNA polymerase makes the new DNA strand, and then these cycles are cycled for usually 25 times. You do these cycles, and then you finally end up with lots of your gene that you've PCR'd up, that you've amplified up. Okay, so there are different conditions that you can change in a PCR reaction. So clearly in your practicals, you're going to be looking at different conditions for annealing temperatures and things like that. And so you have to kind of get to an understanding of why you would use these different conditions. So we often use the term hybridize when you're essentially talking about a primer binding its complementary sequence, and it's very often described as a hybridization, but it's essentially just the association between the oligonucleotide primer, so your primer, and the complementary sequence that you're trying to amplify up. So the binding of the oligo to the DNA is specific to the GC content of the piece of DNA that you're trying to amplify up, and also the length of the oligonucleotide and the salt in the solution. So, and this annealing of the primer to the DNA really only allows for complementary base, a string of complementary bases that don't have any mismatch, and this is what we call a PCR that's done at high stringency, and this would mean that the annealing temperature is quite high in the PCR to make sure that you don't get none of your PCR of your primer binds to bits of the DNA that is kind of only half complementary or something like that to your sequence. If you so some of your PCR reactions in your practical, you will be using higher and lower annealing temperatures and seeing what differences, what kind of products you get, or how different the specificity of the products that you get between a lower and a higher annealing temperature. And so what we expect when it's a low stringency PCR is that the annealing temperature is lower. It means that primers can potentially, the oligonucleotide primers can potentially bind to other regions of the DNA that maybe have some mismatches, so it's not a completely correct sequence, and that way you end up with lots more non-specific products than if you had a higher annealing temperature. That would mean that your primer only binds to the sequence that it's supposed to. Okay, so for example, this is an example of if you use a lower annealing temperature versus a higher annealing temperature in your PCR reaction. So you can see if you use a lower annealing temperature, this is the band of the DNA that you want, so the size. These are your a DNA ladder. You always run a DNA ladder in agarose gels to make sure that you know what size fragments you're looking for. So, okay, so you can see that that the size of the band that we actually want to amplify up is this one. But you can see that if you have a lower annealing temperature, your primers can potentially bind to other places of your DNA template, and you can get other things being amplified up that are non-specific, okay? Then there's multiple bands here. If you increase the annealing temperature, so the temperature that you do one of the steps in the PCR reaction, you can see that you might get less product, but you get only one of the product that you want, okay? So it's one specific single band when you increase the annealing temperature, and the annealing temperature you end up choosing is based on the melting temperature of the oligos that you use. So it depends on the GC content of the primer sequence as to what that annealing temperature is, and we can work that out for all primers. So we should roughly have an idea. You already have an idea before you do a PCR reaction what annealing temperatures you should be using, and then you try out which is the best annealing temperature. So lower annealing temperature is it essentially results in lots more non-specific products. You still get the product you want, but then your primers are binding somewhere else, and the DNA polymerase is busy making other bits of DNA that are not of interest to you. So as you increase the annealing temperature, you get much more specific product of the PCR product that you're looking for. Okay, so a hot start PCR is and we'll be using them for one of our PCR reactions, so not all of our polymerases are what's called hot start polymerases. And a hot start PCR, and this is one of the things you have to also explain in your assignment. So there's various points that you have to go through in the first part of the assignment, and one of them is explaining how the hot start PCR and how the hot start polymerases work. And it's essentially to minimize the polymerase amplifying sequences that are not specific. So that can occur, for example, if you have a hot start polymerase. This means that the polymerase doesn't work until you heat the polymerase to 95 degrees or 94 degrees, which is essentially the start of your PCR. So this first denaturing step heats the DNA and opens up the DNA, but it also essentially activates the polymerase, and it only starts working once it gets to 95 degrees. So you can see that also 95 degrees, this is a protein. So the polymerase, these thermostable polymerases we use in PCR, work at 95 degrees. Okay, so it's a protein in there working at 95 degrees. And again, the only reason why we know we can use these proteins is because all of these proteins that we use for PCR are they were found and are extracted, or now we make them recombinantly from organisms that grow in very hot climates. So, for example, in volcanic hot springs, these kind of areas where it's hot, and that's where the organism lives. And there's lots of bacteria that most of the polymerases are from bacteria that we use, or archaea. So the organism functions at 95 degrees normally, and so we then take the enzyme that normally has no problem functioning at 95, and then we can obviously use it in a PCR reaction. So again, a nice example of tools that we use, just take for granted, but they actually only came because we understood that there were some organisms that like to live in boiling hot springs. Okay, so I touched on this before, and we're going to have a look at this this concept of proofreading when we talk about DNA repair in the lectures after Mark Temple's set of lectures because essentially the function of a DNA polymerase, like the velocity polymerase we're using in the lab, is what's called it has proofreading ability. So apart from making a new DNA strand, which is what a DNA polymerase does, it just copies the template strand, and it's also what's called high fidelity. So it means it doesn't put the wrong base in very often. But if it does put the wrong base in, it checks. So each DNA base that it incorporates when on the growing DNA strand, it checks that it puts the right one. If it hasn't put the right one, it chews away that DNA base. Okay, so it pretty much cuts the phosphodiester bond between that base that it just put in and the one before, and then puts the base on again. Okay, and this is what's called a high fidelity polymerase that has this proofreading ability, and it means it has an exonuclease domain in the protein that has exonuclease activity, and it can chew 3' to 5'. So chew in the opposite direction that you read the DNA. So you're reading the DNA 5' to 3', and that enzyme pretty much chews away the DNA in the opposite direction. So you can remove a DNA base that it's just put on. And as I said, that's also one of the reasons why we use these high fidelity polymerases in cloning, because you don't want to be making mistakes. You don't want the polymerase to be making mistakes if you're trying to put a gene into a plasmid and then use it to do something. But we still do check by Sanger sequencing that we've made the right gene in our plasmid if we're going to use it for something, because polymerases, although they have an error rate, you can see it's about 1 mistake per 10 to the 4 or 10 to the 5 nucleotides. You wouldn't want it to be on your watch that you've made that mistake, and that has happened before. People didn't realise. It's happened a lot as people don't realise they've made mutations where they're not supposed to, and then you're making a protein from that gene sequence, and then you make a mutated protein, and all your experiments are not valid because you're not using the correct protein sequence from that gene. Okay, so as I said, we're going to revisit this idea of proofreading later on in the lectures. Okay, and so we've talked about the fact that exonucleases chew from the 3' end, and endonucleases chew from within a sequence, which are your typical restriction endonucleases. And so this is essentially a summary of what you do in a PCR, in a whole cloning process. You amplify up the gene you want to insert into a plasmid. You engineer ends on the gene using the PCR primers. You then cut your insert as well as your plasmid with those 2 sets of PCR primers that you've chosen. Insert your gene into that vector and then use it for experiments, for lots of different experiments that you can do. Okay, and so the last few slides is going through a few applications of PCR. So, for example, one of the most interesting examples is that you can sequence very old DNA. So, and it's not only Neanderthal DNA that's sequenced. Now we've got all these different hominids, etc. Other Homo species that have been sequenced, and then you can compare. You can do these comparison sequence comparisons between, Neanderthal and look at the origins of modern humans, and etc. So that's obviously a very powerful technique if you can sequence genomes, and that's, as we said, it's a modified version of a polymerase chain reaction. Okay, so obviously one of the main one of the important tools for forensic scientists is molecular biology, and I know that there are quite a few people now. Who here is doing forensic science? Yeah, okay. So anyway, so I know that there's quite a few students doing molecular biology that are doing forensic science, and that's like one crucial tool in the forensic scientist toolkit is to use molecular techniques for lots of different things. So clearly, if you don't have a lot of material, say for example at a crime scene, so you have some DNA, so you have hair, you have something, you can use PCR to essentially make more of that DNA for you to then analyse it and then determine whether, you know, who was at a crime scene based on whether they're in a particular database or something. So PCR is a very important tool in that respect. PCR is really important now in, for example, in IVF, so in vitro fertilisation procedures. So the first thing that occurs in an IVF procedure is you make lots of embryos that are being fertilised, but then they get checked, okay? And so they get PCR screened. So you take a few cells from a developed embryo in a dish, and they get genetically screened, and only embryos that don't have any mutations will then be implanted in the IVF procedure, and that's like used all the time. Yeah. Yeah. So you can determine what you. So yeah. So you. Yeah. So you get a whole genetic screen of an embryo, so you know everything. Yeah. Yeah, so these are the kind of things that you would be looking at in your assignment. Obviously, the whole point is that for the first section of the assignment, that you're explaining PCR and all of these extra things that we talk about in PCR, so the hot start polymerases and all these things, so you have to explain these. As I said, figures are really important to be able to explain what you're talking about. Okay, so I can't highlight that enough, is that figures are really important in these types of assignments where you're trying to explain, you know, methodology or steps and things like that. And the second part of the assignment is where you go and look at your application of PCR. And so what I'm looking for is not just that you said, oh yeah, you can use it in that. Well, I want to know how PCR is used in that application. So what exactly are you PCRing up in whatever application you choose, etc. Okay, so more technical and not just a few lines, oh, it's used in that. Okay, I can find that out too. So I want to know that you've looked for the details, etc. Okay, and so that's it. And so Mark Temple will be the next block of lectures. We'll give those, and then I'll come back for I think the last 4 lectures or something. So, yep.